Okay, students, so let us begin with the same concept that we were discussing for the last class. And in that we have seen that how come the alimentary canal that we have in our body, the mouth and the buccal cavity, the pharynx and the esophagus would look like. And for today's lecture, we would wind up the stomach, the small intestines, and the large intestines. Now, here we have to be very much sure that where does the stomach lies. So as we have said earlier that at the top there would be the mouth and the buccal cavity, and next to that there would be coming up of the pharynx, and then we have this esophagus coming. And after the esophagus, we have got this muscular bag. And this muscular bag was termed as the stomach. And we would see for today that how come the stomach would look like. For the stomach, you have to see is that this is a muscular bag. And one thing that we have to notice is that no matter the buccal cavity that we have, the pharynx we have, the esophagus we have, and the stomach that we have got, so there lies an empty space here. So this means that as long as the food would move along the alimentary canal, the food would only be digested. This is not getting absorbed at the moment. So for the stomach, this is a muscular bag. And if I just try to tell you in case of the stomach, so you see that here we have the stomach wall, and in the stomach wall we would have the gastric glands. Whenever the word gastric is being used, so this is with regard to the stomach thing. So if I were to take you here, so you see this is the stomach wall, this is the stomach wall. At the top we have the esophagus, and after the stomach here there would be beginning of the small intestine. But at the junction between the small intestine and the stomach, we have got this sphincter with us, a band of muscle, and this band of the muscle is the gastric sphincter. But the usual thing that we have to understand in the stomach case is that what we have got in the wall of the stomach. So you see that here is the wall of the stomach, and I will take you into the detail that what we see here. So this is like the part of that is represented here. So this is the bigger diagram that we have to understand. So again, you see this is the wall of the stomach, and the wall of the stomach on the outside, this is protected by a layer, or this is surrounded by a layer, and that layer is a sticky material, and that sticky material would be referred to as, or it would be termed as, the mucus. Now you see that we want a lot of juice to be releasing out of the glands, and we say that in this wall of the stomach we have got some pores with us, and these pores that we see at the moment, so these pores, they are termed as the gastric pits. And these gastric pits at the bottom, they are lined by the glands. Like if you see, I am elongating these gastric pores, and then on the downward, I am making these glands out, and these glands, they are termed as the gastric glands. Now the situation with the gastric glands is that here they have got the cells, and in these cells they would be making up of some juices, and they would be making up of some enzymes that are important for the chemical digestion of the food. So the usual situation that we see is that within these gastric glands, these cells, they would release a juice out here, and that juice, it would be termed as a gastric juice. And within the gastric juice, the enzymes that would be available, so they would not be activated until or unless the gastric juice would come out of the gastric gland and into the lumen of the stomach. Because the scenario is that if the cells of the gastric glands were producing the enzymes in the activated form, so these gastric gland would get digested, and as a result, there would develop some kind of openings or some kind of the holes within the stomach, and that can not be good for our health. So the gastric gland would produce a gastric juice. The gastric juice would have the inactivated enzymes, but the time the gastric juice would come here in the lumen, so those enzymes, they would become activated. So if I were to say these points for you, so we would see that this is the mucosal layer, and this has a sticky substance in it, and that is the mucus. And here what we have to understand further is that here we have the gastric glands, and they are making the gastric juice. And this gastric juice has the digestive enzymes in it, but they would be inactivated. And the inactivated enzyme that we have here in the juice, that is referred to as the pepsinogen. Now the situation is that, okay, finally we have got the inactivated enzymes in the lumen of the stomach. So what would activate it? So that would be the acid present here, and the name of the acid is hydrochloric acid, we say. Now, since the wall of the stomach is living, so there is a quite possibility that this hydrochloric acid can degrade or can digest the wall of the stomach, but this This is prevented by the presence of the mucus because the mucus coming here, this would not allow the gastric juice or the enzymes that they have in it to come in contact with the wall of the stomach. So in this way, our stomach wall would be safe from the catalytic breakdown by the enzymes. Moving further, what you have to remember is that when the food would stay in the stomach for a total of 4 to 5 hours, so here the solidified food would convert into a liquefied form, and that liquefied form is what we refer to this as chyme. Now the situation is that we have got the hydrochloric acid in the stomach. So what would be the pH of this hydrochloric acid and what would be the role of this hydrochloric acid? So the pH of this is very low, that is maintained at 2, and this is such a strong acidic pH we say. And the function that this hydrochloric acid would be rendering is that when we eat the food or when we try to drink the water, so there is a quite possibility that it may have the disease causing organisms in them, and these are the pathogens. And if there was no hydrochloric acid present in the stomach, so this pathogen would have caused some diseases in the human body, but the hydrochloric acid would kill up those pathogens. Other situation was that my stomach gland or the gastric gland, they were releasing the enzyme in the inactivated form, though. So the inactivated enzyme is pepsinogen. So the time the gastric juice would bring the pepsinogen in the lumen, so this pepsinogen would be acted upon by the hydrochloric acid, and the pepsinogen would convert into the pepsin. And this pepsin is actually an acidic enzyme and it also requires an acidic pH. So that acidic pH again would be provided by this hydrochloric acid that we have got with us. Okay, so this is also equally important for providing the acidic pH, and by that the pepsin enzyme that we have in the stomach that have to break the protein so it would get the optimum conditions for working. Now in the mouth we know that the salivary gland they added the salivary amylase enzyme into our food. So if the salivary amylase will remain in the food, so the pepsin enzyme cannot effectively work on the food that we have in the stomach. So what this hydrochloric acid is going to do that it would denature the salivary amylase, so the food can only be acted upon by this pepsin enzyme. And as I have said earlier, that the food would stay in the stomach for 5 hours, and here it would convert into the liquefied form, and that liquefied form is entitled as the chyme. So now you see that when the food would stay in the stomach for total of 5 hours, so this has to move into the small intestine. Now the small intestine that we have in the alimentary canal, so this has got total of 3 parts. Identification or the separation of these structures is not required, but still you may know that the small intestine would have the duodenum, it would have the jejunum, and then would have the ileum in it. And here you see that we have got the diagram of the small intestines coming here. So at the top we had the stomach and this was coming over here, and uh, like time after time the gastric sphincter that we had here at the base of the stomach, so it would relax and the food would pass into the duodenum of the small intestine. So again, if you could just look on toward the small intestine, so here are the intestinal walls with us, like on the both side. And in between that, again, we have this empty space coming, and that empty space would be entitled as the lumen again. And what I see is that in this intestinal lumen or in the wall of the intestine, we have got some minute projections with us, and these tiny projections that I see at the moment, so they are termed as the villi. And we will see in a while that what villi are doing in terms of the absorption and in terms of the digestion. So if I were to show you an enhanced view of this part of the small intestine that lies here, so here you see I have an empty space, and then there would be this intestinal wall, and then finger-like projection are getting out of it, which we are referring to this as the villi. So for the small intestines, there are important, you could say, glands in it, and these glands, they are the intestinal glands, and like the gastric glands of the stomach, they would also release intestinal juices. And these intestinal juices, again, they would have the digestive enzymes that are important for the chemical breakdown of the food. So this part is being magnified here. So you see this is the intestinal wall. On the opposite side, we have this lumen coming. And here, these finger-like projection that I see at the moment, like this. So these are the villi. And these villi further have got some tiny projections more onto them. And these are termed as the microvilli. Now, again, in the wall of the intestine, we have these glands coming. And these glands, they are entitled as intestinal glands, and these intestinal glands will produce intestinal juice, and that would have some enzymes in it. But the situation is that the enzymes that are produced or that are present in the intestinal glands, so they are the free enzymes. Why we call them as the free enzymes? Because we will see in a while that in these villi of the small intestine, we have the enzymes and they are not able to move, but rather than they are strictly placed within the cell membrane of the cells. But the intestinal juice enzymes, they are free. They would be free to move around. Now, the important thing is from the structure of the small intestine we need to have a look on that what villi, how come these villi they look like. So here you see this is a diagram that we have got for the villi. But before I tell you something about the diagram, the situation is that these villi that we have in the small intestine, they would have the cells in them, they are the epithelial cells. Then there would be coming up of the blood capillaries in it. We have the lymphatic vessels, and the other name of these lymphatic vessels is also the lacteals. And in these lacteals, we have a colorless fluid, and the name of that colorless fluid is lacteals. So you have to follow me that how I'm making you understand the diagram of this villi. So from here to here, this is one villi. And on the outside of the villi, if you just follow me, like this part that I'm indicating with the red marking, so this is termed as the epithelial layer. And in the epithelial layer, every individual cell that we have got over here, so this would be termed as the epithelial cell, we say. And the microvilli that I showed you above, so they are actually the extensions that get out of the epithelial cells that we have in the villi. When I would come below the epithelial cells, so here there would be the presence of the blood vessels, and these are the miniature blood vessels, and these miniature blood vessels, they would be entitled as the blood capillaries, we say. And then at the center of these villi, here we have got another vessel, and the name of the vessel is the lymphatic vessel. And in the lymphatic vessel, there would be the presence of the fluid in it, and that fluid would be the lacteal. That would be the lymph. The lymphatic vessels is the lacteal, but the fluid that we have got in these lacteal that is termed as the lymph. Now, in the blood capillaries, blood plasma would be flowing, and in these lacteals or the lymphatic vessels that we have, so there would be the lymph flowing in it. Now, what we need to do next is that I would be making you understand the structure of this epithelial cell, and then we would present a diagram where I would go from the lumen to this epithelial cell, and then I would come to the blood capillaries, and then we would see that what happens here within the lacteal. So let us just try to see that how come this cell that we have got in the epithelial layer, this epithelial cell looks like. If you were to simply see here, so this is the diagram that we have got for the epithelial cell. So this is the cell membrane of the epithelial cell. Down the epithelial cell here, you see I have got this blood capillary with me, and in the blood capillary, there would be the blood flowing in it. And downward the blood capillaries, now here you see that we have got the lacteals here, and in the lacteals there would be the presence of a colorless fluid, and that colorless fluid is termed as the lymph. For the epithelial cell, this is its cell membrane. Okay, so this is the cell membrane of the epithelial cell. And these cells, they also have got some tiny projections coming out of them, and these tiny projections, they are entitled as the microvilli. Other than that, in the cell membranes of these epithelial cells, we have got some enzymes incorporated, and these are the immobile enzymes. These are the fixed enzymes because these enzymes they would only lie within the cell membrane of the epithelial cells, and they are not allowed to move. Moving onward, in the cell membrane of these epithelial cells, again, what we have got with us are these specialized transport protein, and they would be important for the active transport phenomenon, where we would utilize it to be moving the glucose and amino acid into these epithelial cells, and then from here into the blood capillaries. The other situation is that we also have got the abundant mitochondria in these epithelial cells, and these mitochondria they would release a lot of energy in order for the active transport to be carried on with the help of the transport protein. So let me just quickly summarize it up. The situation is that on the outside of the epithelial cells we have the lumen, and in the lumen the food particles they would be broken down. They would also use these enzymes that we have in the cell membrane of the epithelial cells, and from the lumen, like from this empty space, and here we had the lumen, and here we had the lumen. So the food particles they would be moved, with the help of the transport proteins into the epithelial cells, and we would also cause the mitochondria to be releasing the energy out of it. And the enzymes that we have here in the epithelial cells, they would be lactase enzyme, the maltase enzyme, and then we would have the sucrase enzyme. But after we have the food particles here, they would come here, amino acids and the glucose. The fatty acid and the glycerol, they would be absorbed here into the lacteals that we have got. Okay, so this is all what we have to remember in case of the villi. Now, the scenario is that what function these small intestines they would be performing in the alimentary canal. So, in the mouth and in the stomach, the food is partially digested, but in the small intestine, the food that we have got, it would be completely broken down. Okay, like if we are eating the sugars, if we are eating the proteins, or if we are eating the fats, so at then they would be broken down. The sugars would be broken down into the glucose, the protein would be broken down into the amino acids, and the fats would be broken down to the fatty acid and the glycerol here in the small intestine. The other role which the small intestine is rendering, that this would bring about the absorption of the food particles as well. Because as we have seen in the case of the villi, that they have got the adaptations that would allow them to be absorbing the food particles. Now, this is a misconception with the student. They believe that large intestines are the one that absorb the water, most of the water. But this is a wrong approach. These are actually the small intestine that would be absorbing most of the water. So small intestine is important in terms of complete breakdown of the food substances, in terms of the absorption of the food particles, and in terms of the absorption of, you could say, the absorption of the water. Now, if we were to talk about these large intestines that we have with us, and they would come after the small intestine. Now, if I were to present you the diagram, so here we had the esophagus, and then we have got the stomach coming up like this, and then there were these small intestines coming with us, and then onward, when we further proceeded from this point onward, so there were the presence of these large intestines coming, and they would go like this. Okay, the large intestines they basically start from here, and where the small intestine end and the large intestine start. So here we have the blindly ending sac, and that blindly ending sac that we have with us. So this is referred to as appendix. This is the same thing that you hear in your daily life, that we have got the surgery for the appendix. So in some cases, this appendix that lies at the junction of these small intestines and the large intestine, it gets inflamed, swollen up. So the doctor they perform a surgery which is called as appendectomy, where they remove this appendix out of your body. Now, for these small intestine, the first part that we have got for the large ones, so that is the cecum. And from this point to this point, we have got the other part of the large intestine, and that is colon. And this part where the undigested food particles, they would be temporarily stored in the form of a hard form, in the form of the feces. So they would be stored here at the rectum. And then we have got this anus coming. And in the anus, we have again a band of the muscle, that is anal sphincter, that when would relax. So ultimately, we would remove the undigested food material in the form of feces. So if I were to read it up for you, so as if it is written here that the large intestine, they would have the cecum, then they would have this colon coming from here to here, and then we would have this rectum. And in the rectum, the undigested food is temporarily stored. As far as the function of the large intestine is to be bothered about, so like the small intestine, they also have a contribution in the absorption of water and ions. But most water is absorbed in the small intestines, not in the large intestine. And the other thing is that in these large intestine, we have some microorganisms with us. These microorganisms, they work on the undigested food particles, and they make vitamins out of it. So here we are done with understanding the structures that we have in the alimentary canal, and for the next class we will see that how come the physical and the chemical digestion of the food would be brought about by.